Hello and welcome. Today, we're going to connect some dots that might seem pretty far apart. We're going to start with a fundamental law of physics and follow that thread all the way to how we design life-saving drugs. We'll be digging into the really fascinating world of parasite metabolism and exploring how their unique biochemical quirks can actually become their greatest weakness, an Achilles heel that we can target.
So it really does all start here, with the second law of thermodynamics. At its core, it just says that things fall apart. The universe trends towards disorder, towards chaos, entropy. You know, every ordered thing, from a galaxy down to a sandcastle, eventually breaks down. And yet, there's life. Life is this amazing, defiant exception. It creates and maintains these states of incredible complexity and order. And that really sets up the central question for all of biochemistry. How in the world do living things manage to harness energy to fight back against this universal tide of disorder?
So here's our game plan for this explainer. First, we'll look at how life actually wages that fight against disorder. Then, we'll quickly review the standard metabolic blueprint, the textbook version you're familiar with. From there, we'll dive into the really clever adaptations that parasites have evolved. This will lead us right into how we can exploit those very differences for drug targeting. And finally, we'll get into the nitty-gritty of how scientists prove that their new drugs are actually doing what they think they're doing. Okay, so at its heart, biochemistry is the study of how living things create order out of chaos. And they do it by coupling reactions. They take energy-releasing reactions and use them to power the energy-requiring ones that build and maintain all their complex bits and pieces. It's all governed by Gibbs free energy, and for pretty much all life on this planet, that energy at the end of the day comes from the sun, captured by photosynthesis and neatly packaged into molecules like glucose. Right, so let's talk about how cells typically cash in on that stored energy. We're just going to quickly walk through the standard model for aerobic cells. That's the incredibly efficient process that most eukaryotes, including our own cells, use to squeeze every last drop of ATP out of a glucose molecule. And this whole process really rests on three main pillars. It all kicks off with glycolysis out in the cytoplasm. Then it moves into the mitochondria for the TCA cycle. And it all culminates in the respiratory chain, which is embedded in that inner mitochondrial membrane. These three pathways work together like a perfectly oiled machine to break down glucose and capture as much energy as possible. And this diagram gives you a great bird's-eye view of the whole operation. You can see glycolysis happening out in the cytoplasm, breaking glucose down into pyruvate and giving us a little bit ATP. Then that pyruvate gets shuttled into the mitochondrion. Inside the matrix, it feeds the Krebs cycle, which churns out these high-energy electron carriers. And those carriers are what power the main event, the electron transport chain, where the real ATP jackpot is won through oxidative phosphorylation. This really drives home just how important compartmentalization is. In prokaryotes, everything's a bit more jumbled together in the cytosol and at the cell membrane. But in us eukaryotes, the evolution of the mitochondrium was a total game changer. It created this specialized, dedicated factory for making energy. And this physical separation of different jobs is a huge evolutionary advantage, and it's a theme we're definitely going to come back to. All right, so that's the standard playbook. But what happens if you're an organism living somewhere like the human gut, where there's basically no oxygen? The entire respiratory chain we just talked about absolutely depends on oxygen being there at the end to accept all those electrons. Take oxygen away, and the whole system just grinds to a halt. So how do these guys even survive? Well, they evolved some pretty wild alternatives, and I want you to meet the hydrogenosome. This thing is an amazing piece of evolutionary engineering. You find it in anaerobic protozoa like Trichomonas, and we now know it's basically a highly modified mitochondrion that's been stripped for parts. It's ditched its own genome and the entire electron transport chain. It's been completely repurposed for a life without oxygen, making ATP a different way, and, get this, spitting out hydrogen gas as waste. Now, inside this unique organelle, they handle pyruvate completely differently. They don't have the typical pyruvate dehydrogenase complex that our mitochondria use. No, instead, they use enzymes like pyruvate ferredoxin oxidoreductase, or PFOR for short. Now, the key thing to notice here is that they use this unique low-potential electron carrier called ferredoxin. This is a totally different piece of biochemical machinery than what we have. And that difference, well, that's what makes it so exploitable. And that difference, that's our window of opportunity. I mean, this is a central idea behind almost all antimicrobial chemotherapy. You hunt for a biochemical pathway, or maybe just a single enzyme, that is absolutely vital for the pathogen to survive, but, and this is the crucial part, is either completely absent or just very different in the human host. If you can find a target like that, you can design a drug that's lethal to the parasite but harmless to the patient.
Let's look at a couple of classic examples. First up, metronidazole. It's a prodrug, meaning it's inactive until it gets inside the right organism. And what activates it? Those low redox potential ferredoxins we just talked about. They reduce the drug, turning it into a destructive radical that just shreds the parasite's DNA. Our cells don't have those ferredoxins, so for us, the drug is harmless. The second one, which we're going to focus on, is F-flornithine, also known as DFMO. This one targets a pathway for making polyamides, which are essential for the crazy fast replication of trypanosomes, the parasites that cause African sleeping sickness.
And this slide really shows you the sheer genius of its design. On the right, you've got ornithine. That's the normal, natural substrate for the enzyme ornithine decarboxylase, or ODC. Then on the left, you see the drug, DFMO. See how similar they look? That structural similarity is like a Trojan horse. It fools the enzyme into letting the drug into its active site. But the real trick is those two fluorine atoms. Once the drug is bound, the enzyme starts to do its job, but that very process activates the drug, which then latches on and forms an unbreakable covalent bond with the enzyme. It's a textbook case of suicide inhibition. The enzyme literally participates in its own destruction. So, okay, we have a drug that clearly works, and we have a really solid hypothesis for how it works. But, in modern drug development, a good hypothesis is just the starting line. It's not enough. You have to prove it. So how do we actually go about validating that ODC really is the true target of eflornithine?
This is where modern omics technologies give us some unbelievable power. What you're looking at here is a metabolomics heat map. Now, each row represents a different metabolite in the cell, and the columns track what happens over time after we add the drug, from hour 0 all the way to 72 hours. It's like taking a global snapshot of the cell's entire metabolic state. The color coding is pretty simple. Red means a metabolite is building up, and blue means it's disappearing. You can literally see the biochemical chaos that the drug unleashes over time.
And when we zoom in and look at the specific metabolites that are changing the most, the story just leaps out at you. Remember, our drug is supposed to block the enzyme that converts ornithine into putrescine. So what do we see? Well, look at that. Ornithine, the substrate that comes before the block, piles up like crazy. And putrescine, the product that should have been made, just vanishes. This is the biochemical smoking gun. The data lines up perfectly with our hypothesis. But if you want the ultimate proof, the gold standard, you have to turn to genetics. And this is just a beautiful, elegant experiment. Scientists use molecular tools to literally go in and delete the gene for ODC from the parasite's genome. The logic is so simple. If the enzyme is the drug's true target, then getting rid of the enzyme with genetics should have the exact same effect as blocking it with a drug. And that is precisely what happens. The knockout cells stop growing. Then comes the rescue experiment, which is just brilliant. You add back putrescine, the very product they can no longer make, and boom, they start growing again. That proves the effect is specific. And the final nail in the coffin? You inject these knockout parasites into mice, and they can't cause an infection. This confirms, beyond any doubt, that ODC is an essential and valid drug target. So there you have it. We've gone from a fundamental law of physics all the way to the rigorous modern validation of a life-saving drug. And this whole approach, finding and then exploiting these unique biochemical pathways, is still one of our absolute best strategies for fighting infectious diseases. Which of course leaves us with a big question. As we sequence more and more genomes and map out these incredibly complex metabolic networks, what other unique adaptations are out there, just hiding in plain sight, waiting for us to discover them and turn them into the next great therapy?